Welcome to Study Loop.
What happens when you mutate EIF2? Okay, what is EIF2? EIF stands for Eukaryotic Initiation Factor. Okay, so it is an initiation factor that is found in eukaryotic organisms. What does it initiate? It initiates translation. So here, the E's are not drawn in this schematic. They're not written out, but we have the initiation factor 2 right there. The initiation factor 2 binds to GTP. GTP is an energy carrier molecule like ATP, but with G instead of A. The IF2 binds to GTP, it binds to a host of other initiation factors, and it binds to the small subunit of the ribosome. Remember, the ribosome is the machinery that carries out the translation of RNA to protein. So, and that machinery has two subunits, a small subunit and a big subunit. So this is part of the ribosome here. So the initiation factor 2 binds to GTP, a host of other initiation factors, and to the small ribosomal subunit. Once that complex is in place, mRNA and tRNA join the party to then give you this complex right here. So in this complex right here, we have the small. So let me switch colors here, since we're at a new stage. So we still have the small subunit here, the initiation factor 2 here, the tRNA, and the GTP. Once all of this is in place, the big, right, so this is the other ribosomal subunit down here. Okay, this guy now joins the party, and when the big ribosomal subunit comes there, you get the harnessing of that third phosphate from GTP. So that third phosphate gets broken off. So this is where the energy is harnessed in this entire operation. And once that big ribosomal subunit binds, IF2, the initiation factor 2, along with all of the other initiation factors and GTP, they go away. They have done their job. Their entire job was to bring the tRNA, the mRNA, and the two ribosomal subunits together. Okay, so this is the overall process. So a mutation in the IF2 would be detrimental if it prevented any of these interactions. Okay, so now let's look at our answer choices. Choice A, the mutation prevents the initiation factor from binding to RNA. That would absolutely stop translation. If it can't bind to RNA, there's no translation with RNA. The RNA is what is being translated. So that is not our correct choice, because, so again, read the question carefully. We are looking for the one that would not be affected. So binding to RNA would be affected. So that's not what we're looking for. Choice B, the mutation prevents the initiation factor from being phosphorylated. Let's look back at our schematic here. Is there phosphorylation of the IF2 at any point in here? No. The only place that phosphate is involved is when that third phosphate is cleaved from GTP, resulting in GDP plus phosphate. So initiation factor is not phosphorylate. So choice B is the one that would not be affected because that doesn't happen. So it looks like choice B is our correct answer. Let's look at the other two, make sure that they would be affected. The mutation prevents the initiation factor from binding to GTP. That would absolutely affect translation because GTP is what provides the energy. Without the energy, you can't jumpstart the process. Choice D, the mutation prevents the initiation factor from binding to the small ribosomal subunit. And that is also correct because that is also absolutely needed. So if that is affected, this can't happen, which means it's not the correct choice either. So choice B is the only thing that talks about an aspect that would not be affected if the initiation factor 2 was mutated.